Hello. Hi.
Hello, John.
I realized I don't have a waiting room enabled for this. Is that a problem?
I don't think so.
Fine. All right.
So people, they will start coming in, right? You don't have to admit them.
Yeah.
Nice haircut,
No, thank
by
you.
the way.
Just got it a
Suits you.
couple days ago. Thank you.
Okay, looks nice. I'm at home today. I returned from the lab just 10 minutes
Mhm.
ago. So I will have myself muted when people start coming in because I have my dog here.
Oh, good. Okay. Yeah.
And because he's a puppy, he's very noisy. I don't know if you can see him. Like he's
Uh,
messing around with my shoe right now.
nice. Well, okay. That's fine. I've got an introduction ready. I don't think you'll need to unmute. So I say, my name is Jonathan Cannon. I'm an assistant professor at McMaster. I just recently joined the TRF team. We'd like to give some ground rules first. We'd like you to turn your microphone off during the talk. We'll have questions and answers after the talk, unless the speaker suggests something else. And you can ask questions by entering in the Zoom chat or raising your hand. We'd like to thank Dr. Vatakis. Is that how to pronounce his name?
Yeah, that's correct.
Dr. Teki, Dr. Wiener, and the what is the ECR committee?
Early researchers committee
The career
of the
researchers.
TRF. Yeah.
Okay.
Early career, yeah, researchers. Yeah, because we always thank all of them for putting these meetings in place. Somehow we all kind of contribute to these efforts, that's why. And then you move on to the introduction of the speaker, a short biography and his title, and that's about it.
Yeah, I don't feel like I need to give where he went to PhD. I'll just talk about what he's working on and the fact he hosted the second timing research forum, right? Before everything went online.
Okay, I don't have a memory
Yeah.
of that, but
The
you sure definitely
back
know.
when it met in person. Um, yeah.
And also, I will let you know when we know more about the next conference, because I know you're very interested in that as well. I remember our first talk, and you said you're really interested in the organizational stuff. So
Yeah.
Dr. Vatakis knows that already. So when we have something, kind of stable, actually, about the venue and etc. It's going to take place in Portugal, but it's not something that it's ready for advertising. It's not booked yet, so
Well, that's
that's
good to
for your
know.
knowledge. So I asked our hero to add you to all the group emails if you're not there already, so you receive all the updates, etc.
Great.
So she's going to do that because she has access to the group mailing lists.
Yeah. All right. Oh, it's 5 minutes before start time. I'm surprised. We, I mean, did we ask? I guess we didn't ask him to come early to do any kind of check. So
No, because I
guess it's
didn't
fine.
have the contact with him. I
Oh,
said,
right.
let's not jump in to your conversations. But I know that already 106 people subscribed to attend the event. Usually, not all of them are attending, but I think they will start coming in shortly.
Okay. Good.
And I also sent a reminder just a couple of hours ago with the login details. I don't know if you have received
Yeah.
that.
Yeah. I think so.
So
Or
yeah,
yeah.
they are correct. Because I start thinking like, did I mess up the login details? but apparently not.
Yep, and here come people. You can play some background music.
Mm-hmm.
Volume okay?
Everything turn, turn, turn. There is a season, turn, turn, turn. And a time to every purpose under heaven. A time to be born, a time to die, a time to plant, a time to reap, a time to kill, a time to heal, a time to laugh, a time to weep. To everything turn, turn, turn. There is a season, turn, turn, turn. And a time to every purpose under heaven. A time to build up, a time to break down, a time to dance, a time to mourn, a time to cast away stones, a time to gather stones together. To everything turn, turn, turn. There is a season, turn, turn, turn. And a time to every purpose under heaven. A time of love, a time of hate, a time of war, a time of peace, a time you may embrace, a time to refrain from embracing. To everything turn, turn, turn. There is a season, turn, turn, turn. And a time to every purpose under heaven. A time to gain, a time to lose, a time to rend, a time to sow, a time for love, a time for hate, a time for peace. I swear it's not too late.
We're still waiting for our speaker, so everyone, thanks for your patience.
Another turning point, a fork stuck in the road. Time grabs you by the wrist, directs you where to go. So make the best of this test and don't ask why. It's not a question but a lesson learned in time. It's something unpredictable, but in the end is right. I hope you had the time of your life. So take the photographs and still frames in your mind. Hang it on a shelf in good health and good time. Tattoos of memories and dead skin on.
Hey everyone, we don't have our speaker just yet. There may be some confusion about timing, so we'll have to wait and see. Thank you for your patience, and we'll keep you posted.
It's something unpredictable, but in the end it's right. I hope you had the time of your life. It's something unpredictable, but in the end is right. I hope you had the time of your life.
Hi, John. I'm sorry. I was thinking that I have 2 hours.
Oh, no worries.
I was wrong on my timing.
Yeah, no, that's okay. That's okay. It's I mean, timing research forum. We understand that people can have trouble with time.
I'm
One
so
moment.
sorry.
All right.
Well, we can start whenever you want.
All right. Yeah. Well, I think we're probably ready to go. Okay. Everyone, welcome to Timing Research Forum Journal Club. My name is Jonathan Cannon. I recently joined the team helping to organize these sessions. I'm an assistant professor at McMaster University. So first, our ground rules for the talk. Please turn your microphones off and leave them off through the talk. We'll have Q&A after the talk, of course, unless Hugo invites questions partway through. And we ask questions by either in the Zoom chat or by raising your hand. We'd like to thank Dr. Vatakis, Dr. Teki, Dr. Wiener, and the Early Career Research Committee for making this happen. This talk will be recorded and uploaded to YouTube afterwards. So unless there are any other questions, it is my great pleasure to welcome Dr. Hugo Merchant. Since 2004, Hugo's lab at the National Autonomous University of Mexico has been investigating motor areas and motor-related processes in the brains of monkeys and humans. His impressive publication record includes pioneering work on brain mechanisms of timing. He also edited the book on neurophysiology of interval timing. And back when Timing Research Forum was the name of a in-person concert series, or conference series rather, Hugo hosted the second TRF and provided a wonderful opportunity for the Timing Research Forum community as we know it to come together. Finally, I know from firsthand experience that Hugo is very kind and generous with his time towards early career researchers. Hugo, I turn the screen over to you. Thank you so much for joining us.
Thank you. It says that I cannot share my screen. I don't know why.
One moment, it's probably my problem. Let me just check on settings. Oh, hold on. I need to stop my sharing computer sound. How about now?
Okay, so can you see my screen now?
Yep.
It's in presentation mode. So, so we are all set.
Okay.
Well, I would like to thank Jonathan Cannon for the invitation. I really love TRF. I think it's a wonderful organization with a very nice concept. In fact, I organized the second TRF here in Mexico, which I think was a very nice success, although it was a lot of work and I will never do something like that again in my life. And well, I would like to share some of the things that we have been doing in the lab. I think I will prefer that you ask questions at the end, but if you really don't understand something, you can stop me. So the title of my talk is a premotor amodal clock for rhythmic tapping. As you always know, the organisms quantify time over a wide range of durations, ranging from microseconds all the way to circadian rhythms. But in the middle, we have the hundreds of milliseconds scale, which is the scenario of very basic but very important behaviors such as interception and collision avoidance that are present since invertebrates, but also from very complex behaviors such as perception and production of speech, music, and dance. And this is the range where I'm focusing my research. So let's start just by defining beat-based timing as the ability to detect the regular pulse or beat in music and to respond synchronously to the beat during dancing and music ensemble playing. So what I'm showing here is the music score and the auditory input of a very simple rhythmic piece of music. And these pieces of music generate very easily an internal beat perception. And this beat perception not necessarily corresponds to the auditory input, since you can still maintain this internal beat even when there is not an auditory input. And this happens a lot in syncopated music, like in salsa or jazz, where the beat is in silence. So besides the fact that you have this cognitive construction, you also have the urge to move to it. So you normally produce movements that can be taps or head bops that are a few milliseconds before this internal representation, and you can maintain moving with this internal beat even if there is no stimulus at all. In fact, recent studies have shown that the tapping times are very good one-to-one reference of when the internal pulse is occurring, even when you have very complex rhythms. So we can say that beat perception and framing is a very complex cognitive process, and the narrow basis of these cognitive processes are largely unknown. And that's why I dedicated a very large research program in my lab to try to understand what's going on. So for a long time, beat perception and entrainment has been considered specific to humans. However, my lab and other labs have provided a lot of information sustaining that monkeys have the audiomotor machinery for regular pulse perception and entrainment. And in fact, few years ago with my friend Heng-Jan Honing, we suggested the gradual audiomotor evolution hypothesis, which, essentially is suggesting that beat perception and entrainment has emerged gradually in the primate order, so that the large human flexibility to entrain to complex music beats depends on the close dynamic interaction between the auditory and the motor systems. So we know that the dorsal auditory areas of the human brain generates very large connections with the posterior parietal cortex, which in turn also is largely connected with medial premotor areas in the frontal lobe that generate a very strong cortical basal ganglia loop. So we know from functional imaging that all these areas are largely engaged during beat entrainment, but also during beat perception, even without overt movement. So this is a critical circuit for this type of behaviors. We also know in the monkeys that the auditory cortex sends scarce projections to the posterior parietal cortex, which in turn projects largely to the medial premotor areas. And we also know that these medial premotor areas generate a very strong basal ganglia circuit in the monkeys. So what we suggest is that the monkeys have all the audiomotor machinery to perceive and entrain at least to the easiest beat, which is an isochronous beat. So to test this in the lab, we have trained monkeys in different tasks. The actual task is a synchronization task that has two periods. The first one is a perceptual epoch where the monkey places his free hand on a lever, and then we present the metronome composed of very brief stimuli with a constant interstimulus interval. Then after 2 intervals in the perception, the monkey enters the synchronization where it needs to respond in synchrony with the metronome. And it's important that to mention that the time between the taps and the stimuli should be within a window if the monkey wants to receive a reward. It's also important to mention that this is a very dynamic task, because from trial to trial, we're changing the target intervals ranging from 550 all the way to 850 milliseconds, and we can use visual or auditory metronomes. So this is a video of a monkey using an auditory metronome. I'm not sure you can hear it. Can you? Yes? Okay.
It's quiet, but we can hear it.
So let me play it again. So it starts with a, a slow tempo. Next try is a faster tempo. You have the perception and then the synchronization. And then you can see perfectly how the synchrony between the stimulus and the response. Okay, so what you do is you compute the asynchronies, which is the time between the stimulus and the response. And you use a polar representation where 0 degrees is a perfect synchrony between the stimulus and the tap. You can see that the asynchronies are close to 0, and they can even be negative, which means that they are predictive. If you compare them with a task in which instead of having regular stimuli, you have random intervals between each stimulus, what you get is a reactive response with asynchronous that are quite larger than the ones that you observe in the synchronization task. And also, if what you do is you train the monkey to respond even when there is no stimulus, and you compare the asynchronies, you can see that the asynchronous with or without the stimulus, at the end or within sequence is pretty much the same. Now we have trained monkeys in a very dynamic situation in which from trial to trial, we're changing tempos, even during training. And what we got is an amazing behavior. So this is the constant error, which is a measure of accuracy, is the difference between the produced interval and the target interval. So if you have 0 means that you have perfect accuracy. And you can see the distribution is quite close to 0 for a range from 400 all the way to 11,150 milliseconds. So this is amazing. And if you watch the the asynchronies are also quite close to zero. So it means that monkeys can really have proper tempo and phase matching when you train them properly in these synchronization tasks. In addition, in these particular monkeys, we can see the scalar property, which is an increase in the temporal variability of the produced intervals as a function of the target interval. So we can summarize that monkeys can produce rhythmic intervals with proper tempo matching. Macaques can predictively train to an isochronous beat, generating tapping movements in anticipation to the metronome pulse, and that the variability of the rhythmic tapping follows the scalar property. But this talk is mainly a neurophysiological talk. So also a few years ago, we suggested a hypothesis. It was based mainly on functional imaging studies. And this hypothesis suggests that timing depends on two main elements. The first one is main core timing network that is constituted by the supplementary motor and pre-supplementary motor areas, the basal ganglia and the thalamus, and then by a series of context-specific structures. Then when they interact with the core timing network, they define the temporal properties of a particular task. And then what we have done across the years is to focus mainly on the medial premotor areas, which, is the sum of PSMA and SMA here in the medial wall of the frontal lobe. So this is just where we recorded in our initial recordings in a tapping task. And one of the things that we found initially is that the instantaneous discharge rate of neurons show ramping activity. So we found what we call motor neurons, which are neurons that have a very stereotypic response profile independently of the produced interval by the monkey. But we also found neurons that were ramping with longer durations and slopes that were more steep for shorter durations, so that these ramps have the tendency to peak at a particular time before tap. So this is the typical ramping profile of a neuron that is predicting how much time is left for an event. And they have been described in different timing tasks. We found it in this rhythmic tapping task. In addition, we found two types of neurons that what they do is quantify the lapse time from the previous tap instead of predicting what's going on the future. The first one, what it does is that it has an up-down profile of activation whose duration increases as a function of the produced interval. And we have also what we call the sand clock neurons, or time accumulator cells, whose peak of activity linearly is related with how much time has elapsed. And in this paper, we found that there is an interaction between neurons that predict and compute elapsed time. But in addition, across years, what we found also is that the overall discharge rate of neurons is tuned to the parameters of our task. So we found neurons that were tuned for short tempos in our synchronization task. So this is a raster where these dots represent the tapping times. They are aligned to one of the elements of the sequence. And this neuron, has bias towards shorter intervals. This other neuron has bias towards longer intervals, but they also have selectivity for the serial order of a task. It's like the monkey is counting 1, 2, 3, 4, 5, 6, and then you have neurons that encode the sequential and the temporal elements. And here you have the receptive fields for these two neurons. And of course, at the population level, we found neurons that cover all the possible serial order elements and all the tested durations in our task. However, something important is that these tune cells were far from static. If what I'm presenting here is neurons that are tuned to serial order or iteration, and what we are doing here is to stack them, you know, in the x-axis, and we order them based on their peak of activity. And then what you get here is a sequence of neurons that are activated in a sequence, producing a very dynamic change of profile of activation. So to try to understand this, which is a multidimensional type of problem, what we did is to use you know, dimensional reduction techniques to try to understand what was the dynamics behind these signals. So we use principal component analysis and other type of reduction technique, dimensional reduction techniques. But the main goal of these techniques is to characterize how the firing rates of different neurons covary during task execution, providing, you know, a view of shared neural processes captured in these latent variables. So if you have the time varying activity of neurons and you project them in a low dimensional space, you can get latent variables that can create state-based type of representations, which gives you the dynamics of the population in a task. So, and then what I'm showing here is the neural trajectories based on this PCA analysis in a tapping task for the first three principal components, or for the 2 and 3rd components, so 3D and 2D, while the monkey is performing the task. So what you can see is that these trajectories start in a particular spontaneous activity. They reach an orbit for task performance, and then for every single produced interval, they produce a loop. And an important thing of these loops is that there is a section in these loops where the taps occur. Okay, so we published this a few years ago, and what I'm going to focus now is not on this paper, is on a paper that we just pulled up on the bioarchives a month ago, and it's a paper where we try to understand what's going on with these neural trajectories when we compare visual versus auditory metronomes. Okay, so the task is the same. It has a perception epoch where the monkey is not moving, and then a synchronization epoch where the monkey needs to synchronize with these brief stimuli. And if the synchronous are too large, it's not going to receive a reward. If it's within these, you know, error windows, it would receive a reward. And in this case, we use auditory and visual metronomes and only 2 tempos, a short and a long type of. And this is the behavior of 2 monkeys in this experiment. We see that the variability of the produced interval follow the scalar property, but also you can see that it's much more larger, the variability for the auditory than the visual here in red. You can see that the accuracy measured as a constant error is close to 0, which means that they are producing, you know, the intervals that we want they produce. Although again, there is more variability on the auditory condition. And what we see is that there is some sort of error correction type of mechanism in this behavior, since consecutive produced intervals have a negative correlation between them. So it means that if you have a long produced interval, the next one is going to have the tendency to be short. And if this is short, the next one is going to have the tendency to be long. And this can be captured using the autocorrelation lag 1. And you can see that for our 4 conditions, the lag 1 autocorrelation is negative and is larger for the visual than the auditory conditions. So what we can say is that monkeys are much better with visual metronomes than with auditory metronomes, which is the inverse of what you can see in humans. but nevertheless, they have all the fingerprints of rhythmic tapping. In addition, we also characterize the kinematics of the tapping by using automatic video analysis on the behavior, where we computed the speed across the task. And with a threshold, we were able then to identify the movement times and the dual between movement times. Okay, these small dots are the tapping times. And then what you can see is that the movement duration is stereotypic. So the monkey uses exactly the same down-up type of movement. Doesn't matter if it's auditory or visual, doesn't matter if it's long or short. However, if you measure the dual duration, you know, the pause between movements, you can see an increase in the dual for, you know, auditory and visual. So the monkey solving this tapping task by pausing their movements and then producing stereotypic movements during, you know, the sequence. Then we recorded the neural activity. In this case, we were able to record between 50 and 150 neurons every day during task performance using these silicon chunks in the two supplementary motor cortex simultaneously. And the first thing that we saw at the single cell level is that the neurons encoded duration, modality, and serial order. And most of these neurons also encoded, here in blue, elapsed time. So it means that they are multiplexing a series of parameters. And in fact, you know, you can see in the Venn diagrams that the intersections are, the neurons in the intercepts are really large. The mutual information for these parameters is quite above randomness, which is these permuted values in the circles. And most of the neurons encode more than one parameters and were considered nonlinear mixed selectivity neurons. So this is the single cell level. Again, we see these beautiful, beautiful neural sequences for across our four task conditions, where you can see then, you know, an activation where there is a sequential activation of neurons over time, covering all the produced interval, you know, and then there is also some repetitions on the next intervals produced. So again, on these data, we produce, you know, we computed the PCAs, and what I'm showing here are the neural trajectories for our four conditions. You can see that, again, they have a rotatory type of property. You can see again that the amplitude is larger for longer durations than for shorter durations. But you can also see that there is some sort of displacement of the trajectories between the auditory and the visual conditions. So to start to parameterize these properties, the first thing that we did is to compute the subspaces for all these parameters, which is the subspace that explains better a particular parameter. In this case, this is the duration subspace, where you can see clearly that the amplitude is larger for longer durations in the two modalities. You can also see the internal pulse subspace. So in this case, this is the subspace that explains the larger variance associated with the taps, which are these green dots. And an important part of this subspace that is a line is that it's pretty much orthogonal to the duration subspace, suggesting that you have two populations of neurons, one related with tempo, the other one related with generating the internal pulse. When we measure the modality subspace, we saw, around 57 degrees between the visual and auditory subspaces, suggesting that these are sharing, at least they are partially distributed populations between modalities. And we also saw that the serial order subspace really is explaining around 5% of the variability, which means that in this particular task, serial order is not that important. But then what we did is, what happens if we just focus on elapsed time? If we focus only on the elapsed time, we found this amodal clock that is a relative timer. So, every revolution means one produced interval. It doesn't matter if it's short or long, or if it's with visual or auditory cues. And it also has this, what we call separatrix behavior for the taps. All of them converge in a particular space in this PCA space. And what we are hypothesizing is that every time that this relative timer reaches this zone, you generate a pulse. So this is a relative timer, but where is the tempo? So to try to understand where is the tempo, we analyze the kinematics of our trajectories. And especially what we did is to measure the amplitude between some sort of arbitrary point in state space and all the dots in our trajectories. And we get this type of amplitude profile, where you can see that there is larger amplitude for longer durations. So next, what we did is to compute the area under this amplitude for the dual versus the movement in behavior. Remember that the monkeys are solving this task by waiting during the dual and producing very stereotypic movements during the tapping. And then what you can see is that just during the dual, there is an increase in the amplitude. So it seems that the amplitude is a key parameter for the tempo on our task, not during the movement. And another interesting thing is that if what you do is you compute the lag1 autocorrelation on the dual amplitude, you can see also negative values that are larger for the visual than the auditory situation, which is similar to what we observed in the behavior. So this is a narrow correlate of error correction for a synchronization task. On the other side, we also saw that there is a displacement between the visual and the auditory trajectories that can be captured on the position with respect to this arbitrary point, so that you have pretty much the same clock for auditory and the same clock for the visual, but they are displaced. So, and another important thing is that if you compute the variability of the position, you can see the scalar property for this particular parameter. So what we're thinking here is that modality is something that is related with an external output. And this external output is what it does is just displaces the trajectories in two subspaces, but it doesn't touch the clock. So finally, in terms of kinematics, we also measured the speed of the neural trajectories. This is the speed. In blue is the speed for one of our conditions. You can see that there is a large peak at the tapping times. And again, here you have the movement and the dual time based on the movement speed. And the question is how this how the speed of the trajectories are changing as a function of our two main epochs, the movement time or the dual time. And what we found is large peaks across conditions for the movement time, with not a lot of changes in terms of duration, but during the dual, you see a large decrease in speed for longer durations. And this is a fingerprint of temporal scaling, which means that the trajectories are, have changes in speed that also computes time, which is something that has been already, published by different researchers in time tasks. So if we compute then the speed for movement across our conditions, during the movement, there is some modulation, but the larger modulation is during the dual. And of course, there is a decrease in speed for longer durations. So this is telling us that we have amplitude and we have speed modulation. So who's winning to compute the tempo in our task? So to try to parameterize that, what we did is to define what we call the amplitude modulation time scaling index, or AMSI. Okay, and then what this AMSI index is doing is, if you have the scenario of pure temporal scaling, you have perfectly similar trajectories, but changes in speed with longer speeds for shorter durations, and no changes in amplitude. While if you have perfect amplitude modulation, you have no changes in speed, but you have a large change in the radius of these trajectories. And then when we use these on the real data, you can see AMSI here on the bottom part, across our produced intervals in our task, you can see that there is first large time scaling component that increases, and then there is a large amplitude modulation, and then it goes down in a cyclic fashion. But the important issue is that in the dual, we have values in the AMSI equals 0, suggesting then that the computation of time by these neural trajectories is more or less even between the two parameters. In addition, what we did was session-to-session analysis of the neural trajectories and behavior, so that we can have a trial-by-trial type of correlation. And in these sessions, as I mentioned, we have between 50 and 150 neurons, and on each of these sessions, we computed the trajectories, and then all our parameters, and we correlated that with behavior. And we found that the neural trajectories have systematically 3 parameters that are related with behavior. One of them is the amplitude of the dual amplitude of our trajectories that are highly correlated with the produced interval. We also have high correlations between the dual amplitude lag1 autocorrelation with the behavior lag1 autocorrelation. And also we found a very large correlation between the temporal variability of the monkeys and the position variability in our trajectories. And so there is some nice correlation between our properties in terms of the trajectories and the monkey's behavior. So what we are suggesting is a hypothetical mechanism based on a dynamic system approach. So what we have here is the typical, dynamic system equation. And what we are suggesting is that X corresponds to the activity of the studied neural population, and Fx is the timing and represents the interaction between cells in the medial premotor cortex, and U is associated with the external inputs to these areas. And XO is just the spontaneous activity of the neurons. So what we are suggesting is that our complex trajectories depend on 2 components. One of them is this amodal clock that has amplitude modulation and temporal scaling, and that depends on the interactions within the circuits in the medial premotor areas. And we also have, this U component, that is these external inputs that gives you these displacements in trajectories between auditory and visual. And we are also suggesting that there is an anatomical substrate for this. So the amodal clock is in the premotor areas, but this external input depends on the unequal inputs for auditory and visual information to these premotor areas. So we perfectly know that there is a very large connectivity between the visual system and the posterior parietal cortex. And we also know that there is scarce projections from the dorsal auditory areas to the same, areas in the posterior parietal cortex. And then this area 7A sends uneven type of projections, which probably generates these uneven external inputs and our general trajectories. So summarizing, we can say that there is an amodal representation of beat-based timing with two components, the trajectories that converge in a similar state space at tapping time, providing the phasic representation of a cyclic time event or the internal pulse, as well as the continuous and relative lecture of how much time passes within each produced interval using this loop type of representation. The tempo of the tap synchronization depends on the dual between stereotype movements, and this dual is encoded by a combination of amplitude modulation and temporal scaling in the neural trajectories. We can say that modality imprints specific signatures in these neural trajectories with a large displacement in state space without greatly altering the cyclic organization, duration-dependent changes in amplitude and temporal scaling, nor the tap attractor behavior. And these findings suggest that rhythmic entrainment to metronomes with different modalities depend on the interaction between this amodal internal representation of pulse in MPC and the modality-specific external input. So at this time, Jonathan, I'm not sure if I have more time because I have a series of slides that are, I think, pretty interesting. But I'm not sure, that depends on you. Or we can open this to discussion. It's up to you.
Well, I mean, I think we usually leave about 15 minutes at the end for discussion, but I think people are excited to hear a little bit more. So, I mean, do you want to take 2 or 3 more minutes to walk us through a little bit?
Yeah, it's going to take like 10, but yeah, I will try to make my point clear and short. So we have these neural trajectories, okay? But is this clock only neural trajectories? So because you have these neural sequences going on. So I was trying to try to characterize which aspects of these neural trajectories are related with the properties of the neural trajectories. So for that, what I did is to characterize the activation periods of each neuron, using a particular type of analysis that gives you where the activity starts, where it finished, and where is the peak. And we sort again these neural sequences based on the peak time. You can see again, these neural sequences that are repeating each other, generating this regeneration, and where there is at the end of the tap there is some sort of restart of these neural sequences. So with this information, I was able to measure the duration of the activation periods, how many neurons are in each of these neural sequences, and what's the speed of recruitment of these neurons within the neural sequences. And I plotted these parameters as a function of phase for the 4 produced intervals, and this is the mean across all the produced intervals. You can see that the duration of the activation period is larger for longer duration than shorter durations. You don't see a lot of changes in terms of discharge rate. You see large changes in the recruitment lapse, that is a measure of the speed at which these neurons are recruited. And also you see large changes in the number of neurons in the sequence with cyclic changes and peaks just before the tap on the number of recruited neurons. Okay, so another thing that we did is to see how this neural sequence generalized across conditions. So what I have here is heat maps of neurons that have been organized based on the peak of activity for this particular task. But I also use this sorting sequence for other task conditions to see if the order was generalizing. And as you can see, in the diagonal, which is self-sorted type of tasks, you see neural sequences that are pretty well structured. But if you use this sorting order on another task, boom, there is some sort of disordering. So to try to measure this, what I did is to develop a geometric type of approach where I computed the Euclidean distance between, between elements in these matrices. You can see that the diagonal for the self-sorter type of tasks is has a distance of 0. But then what I did is to localize the beams with the minimum distance across all of them. And then you can see that not all of these minimum pins are in the diagonal. Some of them are, all over the place. So I can measure the distance between this minimum distance and the diagonal as a measure of order. Okay, so larger this diagonal asymmetry, larger the disorder between sequences across conditions. And I did this between tasks, between auditory short, auditory long, visual short, visual long, but also across serial order. And you can see that across serial order, this asymmetry diagonal index is not that bad. I mean, there is some disordering as a function of how far you are, for example, from serial order 1 to serial order 4. But this distance, asymmetry is completely different from this one. So when I computed this diagonal asymmetry index, I can see much more larger differences between tasks, which is this matrix, than between serial order, which is this matrix. And an additional thing that I did is to measure this for the beams that are at the beginning and at the end of each interval, or within the interval. And what you can see is that within interval is where you have a reorganization between modalities and a reorganization between durations. Okay, for those beams that are at the beginning and at the end, this asymmetry is not as bad as you would think. So in fact, based on this analysis, what I found is a subgroup of neurons that are active across all durations and modalities. And these neurons occur pretty much at the end of the produced interval. So these cells are some sort of anchoring cells, okay? And it's important because as a final thing, what I'm going to present is some neural sequence simulations to try to see what properties of the neural sequences are related with our neural trajectories. So the first thing that I did, well, we did, was to produce neural sequences that resets for 2 intervals. So you have initial and second interval, they are resetting. In this case, the duration of the activity is the same, but as you can see, there are a larger number of neurons for the longer duration. And what you can get is a longer trajectory for longer number of neurons with an AMC that is typically 1, which means amplitude width. Okay, what you do is simulate neural sequences that have the same number of neurons, but the duration of the activation period is larger. For the larger duration, you get perfect temporal scaling. Okay, this is what you get in many timing tasks. The trajectory is very similar, but the speed changes so that the speed is larger for shorter durations than longer durations, and our AMC is close to -1, sorry. Now, if you start to do a more complex neural sequences, where the first thing that you do is you use the same neurons at the beginning and end of the sequence. So these neurons here are shared in the longer durations here. You get, neural sequence that have these separatrix. They converge at the top. Okay, and that's very important because our hypothesis is that these neurons are the ones that are encoding the internal pulse. Okay, if you add to these neural sequences longer durations for of activation on the tempo that is longer, then you get changes in speed. If at the same time you get larger number of neurons in the longer duration than in the shorter, then you get pretty much the properties that we found in our neural trajectories. So, an AMC that is switching between amplitude and the temporal scaling with values close to 0, in between you have the convergence. And of course, if what you do is you simulate partially overlapping populations, what you get is neural trajectories with degrees that depends on how much the neuron populations are converging, which is similar to what we found between the auditory and visual conditions. So we can emulate pretty much with our neural sequences properties, the properties that we found in the neural populations. So this is an important take-home message. And well, I'm not going to do the summary, but what I want to say is that, of course, the next step is not only record in the medial premotor areas, we need to go and record all over the key brain areas. And of course, some of them are the auditory areas, the parietal areas, the premotor areas, and the putamen areas. And in fact, this is exactly what we are doing now. We already recorded one monkey, and we're starting to record the second monkey, although Juan Pablo, that is listening us now, can tell you that we have some issues with the behavioral computer, and then the experiments are still not running as they should. So I would like to thank especially Abraham Betancourt, which did most of the analysis on the neural trajectories, with Osvaldo Perez, which is a mathematician, is very good, and Germán Mendoza, that is an excellent experimentalist and has helped us a lot with the multi-electrode recordings, and Luis Prado, that is a magician on training monkeys. And also I would like to thank, all the collaborations across the world. And I would like to thank also the funding and say gracias por su atención.
Okay, thank you so much, Hugo. Yes. This is really exciting work. It's so fun to see how you're connecting the PCA results and bringing them back, when connecting them with simulations, and I'm very excited to see where this goes next. I mean, I guess anyone who has to go strictly at 11 should get a question in now. We've got one in the chat. Actually, let's go to Elmira first, and then we'll go to the one in the chat. Go ahead.
Okay, thanks for the great talk. I had a question regarding to the neural trajectories of amplitude and speed in like visual and auditory modalities. I was thinking, like, is it like when you said like the pulse generation in the visual modality is much more accurate, if we look at the neural trajectories of the amplitude and speed, can we say like the separation between these trajectories of long and short interval is a little bit more for the vision? That's why we are more accurate?
Well, I have to say that when we compare on a session by session the neural trajectories with the behavior, the speed was correlated with the produced interval, but it was more often correlated. The amplitude of the trajectories were more correlated with behavior. I'm not sure if I understood properly your question.
Yeah, I mean, like looking into the trajectories, I felt like it's there is more separation between long and short intervals for the visual modality than the auditory.
That's
yeah,
right. That is right. That is right. There is more changes in the overall. You can see it here. That is the raw. Well, you can see, the amplitude is, there is more separation for the auditory than for the visual. That's your point?
Oh, I thought it's the reverse for the other plots, not this one. Yeah, but actually for the speed, it was much more visible as far as I remember.
This is, okay, so this is for the amplitude, and in the case of the speed, this is what you have, right? So this is your point. This is a lot, there is a larger difference here than here.
Yeah, exactly.
Yeah, well, maybe there is some preference in between the two modalities in terms of amplitude and speed, and I don't remember from the top of my mind what statistics is saying in that direction. Sorry.
Okay. Yeah, no, thank you so much.
Thank you. We've got a question in the chat from Iran. Pardon my Spanish pronunciation. Hola, Hugo. Greetings from Uriangato, Guanajuato.
I thought he was in NYU.
A more conceptual question. We see many peers refer to negative values, i.e. asynchronies or lag 1 correlations, as indicators of error correction mechanisms. My question is, if these values indicate error correction mechanisms, why do they persist as negative and don't go to 0 instead, or a distribution clearly centered around 0? What are the neural underpinnings?
Wow. Well, I think that's a tough question. I mean, the lag 1 autocorrelation with negative values has been taken as a measure of error correction in synchronization tasks. However, I don't think it is the non plus ultra measure for error correction, because I think there is other parameters going on. But the point is, nevertheless, that there is a dynamic change within a sequence, so that the subject doesn't lose. If he goes short, the metronome is just far away. You're not going to get it. In the case of the monkey, the trial is aborted because the asynchrony is too negative. So this is one thing that I want to say. The other thing that I want to say is, in humans, you see clearly better lag-1 autocorrelations for auditory metronomes than visual, which is the contrary of what we see in the monkeys. Again, suggesting that monkeys have the bias towards the visual, while humans have this bias towards the auditory. Besides that, and how deep our data are digging into the autocorrelation mechanism, the only thing that I can say is that the only systematic parameter in the session analysis that we saw that it was related with the behavioral autocorrelation was the amplitude in the dual lag-1 autocorrelation. So, which probably then is the mechanism that is used then to say, well, if this was short, the next one needs to be long, because if not, the metronome is going to be lost.
Thank you. So I think I saw a hand earlier. Dobry, if you're still interested in asking a question, you can jump in. But otherwise, I've got a few questions, and then I guess we should wrap up since we're over 11. Let's see. Well, so I have one specific question. You showed a picture of the amodal relative clock where everything was overlapping. How did you isolate that from all the other dimensionality of the neural signal?
So what we did is to find the subspace where only elapsed time was represented by averaging all the other conditions and getting, the eigenvectors and that could project, these mean trajectories into this space. This is how we computed.
Okay. Nobody has a question. Hi, Hugo. For a linear dynamical system, you can expect that you can easily decompose the parameters and map them to behavioral parameters. In a nonlinear system, that's not so obvious. How do you know that things such as amplitude and velocity of the oscillators are the best to work with?
Well, this is simply a correlational type of analysis, and this is what the data are telling us. Of course, you could try to solve this problem using, a more formal approach, but this is data-driven type of analysis where, we have this data where we know that there is part of the clock, and these are the main properties related with tempo, at least during the dual, and this is as far as I can go.
Thanks. So I've got one or two more questions, and then I guess we can wrap up unless anyone else has some questions. One of mine is, you've got these serial order sensitive cells. Have you tried any longer tapping tasks? And in those, you would imagine that you'd eventually reach a steady state of some kind and lose that serial order representation?
Well, in this particular task that I show you, and if you remember, the serial order subspace is explaining 5% of the variance, which is really low, which means that, it's pretty much stereotypic across the sequence. In other tasks, like the synchronization continuation task, that's totally different. We have very long and very large effects on serial order, and then you can see the stack, of the circular trajectories doing some sort of elliptic type of arrangement. So I think that's dependent, number one. Number two, we can push the monkeys to do longer tapping trials, but it's difficult because if you are checking for the asynchronous every time, then the monkeys get an error in the 5th, in the 6th, in the 7th tap, and it's very frustrating for them. So we have not computed trajectories for very long sequences because of that.
Right.
And probably that has to do exactly with the fact that the error correction system in monkeys is not as good as in the human.
Okay, and then one last question. Have you considered or tried or piloted anything, any kind of phase perturbation experiment where you see monkeys actually correcting for an error induced by a shift? Or would that be too frustrating?
Yes, we already did the experiment, and my graduate student that did that experiment is now in Caltech, and he's not here, I guess. He's not here, but he's very busy with our experiments. So we're starting to analyze the data, and they are beautiful.
Oh boy.
Because you can see that the neural trajectories change their properties very dynamically, very nice, depending on the perturbation. So that's as far as I can say, but we have done that. And hopefully next year we will be able to publish it.
Wow, I'm looking forward to that. Well, thank you everybody for attending. I hope you can join us December 14th. Benjamin Morion will be presenting. Have a great afternoon or morning or evening, depending on where you are. And I'll see you next time.
Thank you. Thank you for watching, and thanks for the invitation.
Thank you so much, Hugo.
Bye.